All right, let's dive right in. Today, we're talking about the absolute bedrock of bioinformatics. Seriously, before you can even think about analyzing DNA or writing code to cure diseases, you have to get this stuff down. We're talking about Domain 1, the foundational sciences.
And that's really the million-dollar question, isn't it? We're living in an age where we can generate just an unbelievable amount of biological data. We're talking entire genomes, all the RNA messages in a cell, the whole shebang. But all that raw data, it's just noise. The real magic, the real challenge, is figuring out how to turn that massive jumble of A's, T's, C's, and G's into something that actually means something.
Exactly. The bottleneck isn't data collection anymore, it's data comprehension. And that's what this is all about. We're going to break down the foundational sciences, the theoretical ground floor that the entire skyscraper of bioinformatics is built on. You've got to know this stuff because without it, even the smartest computer algorithms are totally useless.
So where do we start? Well, think of this foundation as being held up by two giant pillars. You need both of them to be solid, or everything you try to build on top will just come crashing down.
And here they are. Pillar number 1 is all about biology. The actual rules of life. You have to understand the system you're trying to study. And pillar number 2, that's about the language we use to make sense of that system. Math and statistics. These are the tools that let us pull a clear signal out of all that biological noise.
So these two pillars give us two core modules. Module 1, molecular biology and genetics, is the what. What is a gene? How does it actually work? How do we even get our hands on this data? Then you've got Module 2, mathematics and statistics. That's the how. How do we organize it? How do we model it? And maybe most importantly, how do we know if a discovery is the real deal?
Okay, let's tackle that first pillar, the biological blueprint. This module is all about the operating system that runs inside every single one of our cells. If you can't speak this language, the data is just going to look like gibberish.
So, right at the heart of it all is something called the central dogma. Sounds intimidating, but it's not. Think of it like this. DNA is the master blueprint for a building, locked away safely in the architect's office. Now, you wouldn't take that whole master plan to the construction site, right? You'd make a photocopy of the page you need. That copy is RNA. Then the construction crew uses that RNA copy to build something useful, like a beam or a window. That's the protein. That flow from DNA to RNA to protein, that's basically the core process of life.
But a gene isn't just one long string of text. It's got grammar, it's got structure. The parts that actually have the instructions for making a protein, we call those exons, but they're often broken up by these other non-coding bits called introns, which get snipped out. Before that, you've got a promoter, which is like the start button for the gene. And at the end, you've got UTRs, which kind of help manage the whole operation. You have to understand this structure to read the data correctly.
Now, this is where things get really cool. The DNA sequence itself isn't the whole story. There's this other layer of control sitting on top of the DNA. It's called epigenetics. For example, your cells can stick a little chemical tag on the DNA, that's DNA methylation, and it basically acts like an off switch for a gene. Or they can change how the DNA is wound up, which acts like an accessibility switch. It's like having the same library of books, but some are locked in a vault while others are sitting right on the front desk. So how do we even read all this stuff? Through sequencing machines, of course. And here's the kicker. The machine you use totally changes the data you get. A technology like Illumina gives you millions and millions of tiny, super accurate snippets of DNA. But then you have something like Nanopore or PacBio, which gives you these really long pieces of DNA. They're great for seeing the big picture, the overall structure. Knowing how your data was made is always step one.
Okay, so we get the biology, but how in the world do we deal with the messy, noisy, gigantic data sets that come out of these biological systems? Well, that brings us to our second pillar, the language of data. This is where we learn how to turn all that noise into actual knowledge.
Just try to picture it. Data from 20,000 genes across hundreds of different patient samples. It's just a huge, tangled mess. Linear algebra is the tool we use to bring order to that chaos. We can organize it all into a nice, neat grid called a matrix. And then, and this is the cool part, we can use powerful techniques like SVD to find the most important patterns hidden in that grid. It's how we boil millions of data points down to one clear picture.
Let's be real. Biology is messy. It's not perfect. There's a lot of randomness. Probability theory gives us the framework to handle that uncertainty. For instance, when you're counting how many times a gene is turned on, the data is really noisy.
It doesn't fit a nice, clean bell curve. So we have to use special models, like the negative binomial distribution, that are built for that kind of messy data. And we use ideas from Bayesian statistics, which basically let us update our beliefs and get more confident as we gather more evidence.
And this leads us to maybe the single most critical idea in this entire domain: statistical rigor. Look, if you test 20,000 genes to see if they're different between two groups, I promise you'll find hundreds that look interesting just by pure random chance. This is why we have to control for the false discovery rate. It's a way of asking, okay, of all the things I'm calling a discovery, how many are probably just a fluke?
To do this, to avoid chasing ghosts, we use something called multiple testing corrections. It's just a way of being way more strict about what we call significant when we're testing thousands of things at once. There are specific methods for this, like the Benjamini-Hochberg procedure, that are designed specifically to keep that false discovery rate low. It's like a super smart filter that helps separate the real gold from the fool's gold.
Okay, let's just zoom out for a second and appreciate the foundation we've just laid down. We've gone through the absolute must-know theories from both biology and math that form the bedrock of this entire field.
This is a perfect summary. You've got the two sides of the coin. On one hand, you've got the what, the biology. We understand the basic process, the central dogma, we know the structure of genes, we know how they're regulated, and we know where the data comes from. Then on the other side, we have the how, our analytical tools. We use linear algebra to organize the chaos, probability to model the uncertainty, and statistical rigor to make sure our conclusions are sound.
And that foundation, well, it's everything. Every advanced algorithm, every complex analysis, everything you'll ever do in bioinformatics is built on top of a solid grasp of these core ideas. You now have the conceptual toolkit you need to move on.
So, what's coming up next? With this theoretical foundation firmly in place, you are officially ready to start building. In our next explainer, we're going to jump into Domain 2, where we take all this theory and finally start putting it into practice. This is where the fun really begins.